This lecture is being brought to you in part by the generous gifts of these sponsors.
All right, well, good evening. Thank you for the kind introduction, Dr. Ford and colleagues, for the invitation to visit this lovely facility and also spend some time with you this evening talking about immunotherapeutic approaches to treating cancer in general, and specifically some of the efforts in trying to tackle one of the most aggressive and challenging malignancies that we deal with in oncology, which is brain cancer. So this is my first visit to the IHMC here in Pensacola, and you guys have a lovely, lovely town. It's really been a pleasure to spend the day here, and hopefully we'll have an engaging presentation and, more importantly, discussion afterwards. I'll try to fill you in on some of the highlights of the field of cancer immunotherapy and some of the challenges and efforts that we have in research to try to make this treatment approach for refractory cancers applicable to more patients in general. Make sure I have this correct. All right. First, in way of disclosures, I do have technologies in the immuno-oncology space that have been patented and licensed by pharmaceutical companies. They are not going to be the major focus of this overview presentation this evening, but I will touch on some experimental therapeutics, which are intellectual property of the University of Florida and co-inventors. So outline, I'll discuss a little bit of history of cancer immunotherapy. This is a field that has received a lot of attention, particularly over the last 5 to 10 years. Most people, if you watch any television, you've probably seen commercials advertising cancer immunotherapeutic treatments for lung cancer, and in particular melanoma and others. But this has really been a field that has evolved for over a century of efforts to try to figure out how to use the immune system to actually recognize cancer as a foreign invader in the body and effectively eliminate malignant cells. I'll talk about the really conventional approaches to targeting tumors using immunotherapy, particularly those that have already received FDA approval for one or more cancers. And while there's a lot of investigational approaches still under development, you'll probably see that this has been a field that just in the last 10 years has actually moved from a research effort to now one of the most dominant forms and effective forms for treating a wide variety of malignancies today. And then I'll talk about our efforts in brain tumor immunotherapy, which is a disease that is not yet seen as a whole, the tremendous benefits of immunotherapy that have been realized in other cancers, and our efforts to try to understand why, where are some of the challenges and opportunities for breakthrough in treating malignant brain tumors. So cancer immunotherapy, how do we get here? As was mentioned in my bio, I studied for my Ph.D. immunology at Duke University starting in 1995. And at that time, I was interested in cancer immunology. I ended up doing my dissertation work in trying to understand how the antigen-presenting cells of the immune system recognized and presented antigens from cancer to the T cells of the immune system. And I ran into an undergraduate student not too long ago who had been had become recently interested in cancer immunotherapy. And when she asked me how long I had been studying the field, I mentioned to her that I got into it in 1995. She said, "Wow, you've been at it since the very beginning." And I said, not really. The history of cancer immunotherapy predates my graduate studies, but it just highlighted really that how long does it take for an overnight success to really become publicly aware? It's about 100 years. And so if you really look historically, the concept that the immune system might recognize and fight cancer dates back at least to the late 1800s. Dr. Coley actually read about a patient who had had a surgical resection for cancer and an incomplete resection, but subsequently a postoperative infection where the wound had become infected with a bacterial infection, and the patient subsequently had spontaneous remission of the remaining cancer. And so he reasoned that the immune system must have been awakened by the actual infection and then recognized the remaining cancer cells and led to an effective rejection, and began a series of actually experiments of trying to purposefully induce cancer regression by infecting patients initially with live bacteria and then some of the heat-killed components of mycobacteria known as Coley's toxins to try to induce whatever this inflammatory or immunologic response that could hopefully spread and awaken to recognize cancer cells. While this approach wasn't largely effective in the majority of patients, some actual lessons that were learned even from the late 1800s in this approach are still used today. And in fact, a treatment of superficial bladder cancers is actually used as a form of derived mycobacterial products to induce an in fact inflammatory response that can, in the case of bladder cancer, actually mediate effective rejection. So the underlying principles have been around for a long time, and subsequently, our understanding of the basic components of the immune system and the steps that are required for effective immune engagement have really led to an evolution now of understanding, at least in some cases, how to either induce an appropriate response or overcome the obstacles that cancer presents to the immune system in recognizing these tumor cells as foreign invaders. One of the seminal breakthroughs in the field and understanding of immunology in general and in cancer immunology has been the recognition that the immune system itself really responds to cues and signals from really commanders or sentinels of the immune system that tell our bodies what really is a foreign invader, what's something that's dangerous versus what is something that we perhaps should ignore or even repair. So the concepts of the immune system really recognizing self from non-self has really evolved to understanding that our immune systems really recognize antigens or challenges in the environment in a certain context. And so you can imagine if you have an injury where you need to both defend against infection but also repair a wound. These are signals where the body can either be feeding the environment with nutrients, blood vessels, angiogenesis in the form of wound healing and repair, or in the setting of an infection really being cued to destroy and kill infected cells and other foreign invaders in that environment. Cancer, in some ways, in a simplistic sense, sends the body mixed signals. There are abnormalities in a growing cancer that the body definitely can recognize as abnormal. There are also foreign proteins or mutations or changes in cancer cells that do elicit proteins that are different than what the immune system normally encounters in healthy tissues. And yet at the same time, many of the signals around injury and repair are actually also triggered by cancer cells. And so the overriding physiological response can sometimes be to feed and grow and try to repair that injury as opposed to recognizing this as something that should be rejected. Ralph Steinman and Dr. Cohen and colleagues discovered the sentinels of the immune system called dendritic cells are antigen-presenting cells that really provide context to the immune system and say that this is either a protein or an abnormality that's dangerous and should be attacked by the immune system, or in fact this is self or a non-dangerous signal and really should either be ignored or repaired. And so dendritic cells ultimately are somewhat like the generals of the immune system army that are either telling the overall immune system to attack or to ignore or retreat. And so with recognition of the really important role of dendritic cells and antigen presentation for triggering an effective immune response, much of our understanding of both vaccine development and therapeutic cancer immunology really centers on having these antigen-presenting cells send the appropriate signals to the soldiers of the immune system. The first real breakthrough in the cancer immunotherapy field in terms of an FDA approval is actually taking advantage of this understanding of the role of antigen-presenting cells. It was actually a cell-based, dendritic cell-based therapeutic treatment for castrate-resistant prostate cancer. In this treatment, the actual antigen-presenting cells are collected from the blood of patients in a large volume leukapheresis. White cells that contain these dendritic cells are actually enriched from this peripheral blood draw or large extraction of white cells from the body. They are actually mixed in a ex vivo culture system with a overexpressed protein that's expressed in many prostate cancers, tumor cells called PAP, and along with a signal from GM-CSF, a cytokine that really activates these antigen-presenting cells. And then they are infused back intravenously into a patient, receiving back their own cells that have now been loaded, if you will, with this tumor-associated antigen. This was actually shown in a randomized clinical trial to actually improve survival compared to chemotherapeutic treatment. It was not a overwhelming miraculous cure of advanced prostate cancer, but was heralded the first FDA-approved immunotherapeutic treatment in the field. It also highlighted some of the challenges. This has not been widely adopted in terms of a standardized treatment, and in fact, many of the challenges of this manufacturing workflow limited its adoption and its ability to turn these cells back around and deliver them back to patients in a timely fashion. After the approval of the cell-based therapeutics for prostate cancer, the real breakthroughs in the field came in 2011 and really over the last 10 to 12 years. And that's been with the understanding that the immune system actually has both activating signals and, importantly, regulatory signals or brakes on the immune system that we call checkpoints that actually tell the body not to respond or not to attack. And these immune checkpoints are oftentimes overexpressed in cancer patients and upregulated on cancer cells, such that any signals that are alerting the immune system that there may be an abnormality here are being overridden by these immune checkpoint signals that are telling the immune system this is self, not to attack. These are very important in preventing autoimmune disease, very important in limiting the duration of an immune response, where we don't want a chronic immunologic response that is not turned off. But in the setting of cancer, these signals really essentially function as a stoplight that's always on. The development of immune checkpoint inhibitors, or the ability to block these negative regulatory signals, has actually been the largest shift in our understanding of the balance between immune rejection or ignorance when it comes to cancer. And these monoclonal antibodies, the first anti-CTLA-4 are called ipilimumab, and anti-PD-1 called nivolumab, and now there's also pembrolizumab. These antibodies do not directly attack cancer cells or directly stimulate the immune system, but instead simply remove the brakes that the patient has on their own immune response and unleashes essentially a more unchecked immunologic response against cancer. These have had dramatic effects in patients with metastatic melanoma, non-small cell lung cancer, and actually now over 15 different cancers and over 32 FDA-approved indications in which immune checkpoint inhibitors are used to treat advanced disease. In 2013, cancer immunotherapy was heralded as the breakthrough of the year by Science magazine. In the subsequent next 4 years, both the American Society of Clinical Oncology as well as the American Association of Sciences had highlighted breakthroughs and advances in cancer immunotherapy as the most significant, or certainly in the top 5 to 10 breakthroughs in the cancer field during those subsequent years. And so this has really been a revolution of 100 years ago, the concept of trying to activate the immune system, to now we actually have metastatic cancers such as lung cancer, where immunotherapy may be the first recommended treatment for patients that are diagnosed with certain forms of advanced lung cancer. And so that's been quite a dramatic shift. This is a list of the cancers with FDA-approved immunotherapies to date. It's an expanding list that, quite honestly, every 6 to 8 months, either a new type of cancer or another indication within these systemic cancers are approved by the FDA. Number 15 is actually a really interesting approval because it represents the first pan-cancer FDA approval for a treatment to treat any cancer that actually has a certain type of genetic alteration. And so these are called microsatellite unstable or mismatch repair deficient cancers, MSI or MMR, DMMR cancers. What this really are patients who have an abnormality in their cancer cells where they are not able to repair the DNA effectively. It turns out that these cancers, because the DNA repair or the stability of the genome is very unstable in those cancers, they accumulate very large numbers of mutations. And it turns out that some of these mutations end up changing the proteins of the cancer cell in such a way that the immune system can actually recognize those mutated proteins as foreign. And so you can use checkpoint inhibitors for these cancers that have these very high levels of mutations. We, in the immunologic field, call them neoantigens. They basically translate into new proteins that have not been seen by the immune system before. And in many cases, regardless of the cancer type, you can achieve effective immunologic responses and clinical responses using checkpoint inhibitors for those patients. So this is the first time a FDA has approved a treatment essentially for any cancer based on simply the underlying genetics of those cancers. And I think it heralds really two huge areas of advance. Obviously, our understanding of the underlying genetics and genomic biology of cancers and how important that has become and how we treat them today. But then also a real signals a huge advance in the use of the immune system to recognize cancers. In our own field of brain cancer, you know, this is not on this list yet, These are cancers that don't typically have the large numbers of mutations that lead to this immune recognition, and so we look at other strategies to try to induce effective immunologic responses. But a small percentage of patients with brain cancer actually do have these underlying genomic disorders, and those patients actually do respond to immunotherapy. And so this has been a real significant advance. So this is a simple understanding of how does the immune system really recognize and kill a tumor cell. Your T cells of your immune system each carries a unique receptor that can recognize your antigen-presenting machinery called MHC molecules, and what they present are, supposed to look over here, they present the proteins that are made inside of any cell in your body, any nucleated cell in your body, presents little pieces of the protein on their surface. If these are normal proteins and are sampled all the time, this can lead to either being ignored by the immune system or even tolerance, because the signals tell the immune system these are the normal proteins of the cell. But when you have an abnormal protein, such as a mutated protein within a tumor cell, or a protein that isn't normally expressed being turned back on in those cells, and importantly, the appropriate cues that tell the immune system that there is a dangerous presentation occurring in this context, now these T cells can recognize antigen and elicit a program where they essentially lead to the specific killing of this target cell through recognition of this antigen. This involves poking holes in the surface of the cell using molecules called perforins, and then injecting enzymatic proteins called granzymes that actually lead to the degradation and apoptosis of that cell. This works very effectively against infected cells, and the goal of cancer immunotherapy is to try to get this to work as effectively against tumor cells. One of the real powerful and always attractive features of the immune system is that this is a very specific interaction. You can see it's almost like a lock and key type recognition, which makes the immune attack very specific. And it also can lead to what we call immunologic memory. This T cell, or a certain subset of these T cells, can remember that they have been activated. And essentially, if they see these same antigens again, can become engaged to even more rapidly kill. So the ability to specifically kill cells, to remember what those cells look like, or what the at least foreign components of those cells look like, and potentially have long-lived immune protection, has always been one of the really attractive features or the potential for cancer immunotherapy. This is an actual picture of tumor cells, of a tumor cell in the center being probed by T cells, and what are highlighted in red are these perforins that you can see are being, these are the granzymes that are being polarized. This cell, you can tell, has actually recognized something on the surface of this tumor cell that is leading it to attack, and it is now polarizing those granzymes to be injected directly into that tumor cell to lead to its killing. And so if there were a normal cell sitting right adjacent to it, these immune cells could kill this tumor cell, ignore a normal cell next to it, and look again for another tumor cell to mediate its specific attack. So this is something that has always attracted people in the field to try to harness that capability to treat invasive tumor cells. Our understanding of why this goes wrong and where we can intervene to try to make the immune system more effective in treating cancer has really led to the breakthroughs in the field to date, and certainly to ones that we'll have in the future. We understand now that for an effective immunologic response, there are several steps that all must occur with the appropriate engagement of specific signals. First, you have to have tumor cells that are expressing tumor antigens. We believe that most cancers that we have looked at do express one or more protein that can be recognized by the immune system as foreign, at least potentially. But these antigens have to be released, typically from dying tumor cells, and reach antigen-presenting cells called dendritic cells in a context where these dendritic cells recognize that there is an abnormality that I want to signal immune attack against. And so if there's the appropriate cues, these dendritic cells pick up these cancer antigens, they migrate to lymph nodes where the T cells of the immune system are circulating. And when that appropriate engagement that I showed in the prior diagram occurs, they can activate these T cells, which then clonally expand into very large numbers, leave the lymph nodes, and have to traffic back into the tumor microenvironment looking for the proteins that they were activated against in the lymph nodes. If they find cells that are expressing those same proteins, they will then lead to that very specific killing that I described. And that will lead to the release of more antigens, and again, a feed-forward mechanism for this cycle to be amplified and to continue until the burden of antigen is decreased or gone, and then this cycle will cease. So in the course of an infection, this proceeds and typically through to resolution of that infection, but obviously in the course of a growing cancer, one or more of these steps is interrupted so that we do not complete this cancer immunity cycle. And really the goals of the different forms of immunotherapy are to intervene at one or more of these steps to try to complete this wheel, if you will, of a physiologic host response. If we look at the forms of immunotherapy that have actually, at least in one or more cancers, been shown to be effective and have reached FDA approval, we really have three what I would call classical approaches and a fourth which has become recognized as a form of immunotherapy that still embody the FDA-approved treatment approaches that we have to date. Sorry, let me go back. That's a pretty impressive immunotherapy response. So cancer vaccines, this is trying to stimulate the immunologic response where we present the antigens that are expressed inside of tumor cells to the immune system in a more effective manner. This can be done by actually loading dendritic cells directly with antigen. I'll show you some studies where we have done that in patients, or delivering the actual antigen directly into the patient so that their own dendritic cells can pick up those antigens and ideally more effectively engage the immune system. Adoptive cellular therapy bypasses those steps of activating the immune system in the body and says, let's extract these killer T cells, or in some cases other immune cells that can kill cancer, grow them to very large numbers outside of the body, activate it in a way that they recognize the patient's own tumor, and then deliver these killer T cells or other natural killer cells back to the patient in the hopes that now they can go find tumor cells and kill them. I mentioned to you the immune checkpoint inhibitors. Think of that as taking the brakes off the immune system, sort of like when you're sitting at a red light and it hasn't changed for a long time. You just kind of blow right through it, right? That's what an immune checkpoint inhibitor does, and it really allows the patient's own response, if there are antigens that are being engaged, to proceed in a more unchecked fashion. And then the last form of therapy, which has at least been approved for the treatment of melanomas, actually uses viruses that have been modified to kill tumor cells, so that they don't infect normal cells, but they replicate inside the tumor. They were originally designed and still have as a goal the ability to spread through the tumor and kill the tumor cells as they lyse the tumor and spread to other tumor cells. But what they've found is that the immune response against these oncolytic viruses usually shuts the actual viral infection down pretty quickly. However, the antigens that have been released from the tumor cells during the course of this response, and the fact that these viruses signal a very strong immunologic context, can now lead to an awakening of an immune response against the cancer. And at least in the case of metastatic melanoma, has been also a form of effective treatment. This is just a pretty visual demonstration of the power of cancer immunotherapy. One of the pioneers in the field, Dr. Steven Rosenberg, who is the director of the surgical oncology branch at the National Cancer Institute. He has been working for many years in trying to treat aggressive melanomas using the immune system, and particularly approach where they harvest the T cells that are actually at the tumor site. He and others had hypothesized that the reason those immune cells are probably found inside the tumor is they are attempting to fight the cancer, but they're obviously not doing it effectively. He has taken those cells out, grown them to very large numbers in an activated fashion, and then returned them to patients with metastatic melanoma, along with cytokines to help those T cells grow in vivo. This is showing a patient that has a huge involved metastatic, probably lymph node in this case, or subcutaneous growth that's just tremendous in size. And you can see after 12 days after the infusion of these tumor-infiltrating lymphocytes or TIL therapy, this patient had a complete response that's been durable for many, many years. And so I actually read Steven Rosenberg's book in 1993 before starting graduate school. He, even back then, had a few examples of patients with melanoma that have responded like this to immunotherapy, and was one of the key influences in my interest in pursuing this field, was this really dramatic demonstration that the immune system really can, if appropriately engaged, treat cancers. But if we look at, with all of these FDA-approved indications now, and the real advance of immunotherapy as a standard treatment for many malignancies, we still recognize that there's a large fraction of patients that are not deriving benefit from these treatment approaches, even in the diseases where immunotherapy has been approved. And this graph really kind of shows, is a simplistic picture of what really the current status of the cancer immunotherapy field looks like. For many refractory and metastatic cancers in the past, let's take melanoma or non-small cell lung cancer, if you treated these advanced cancers at what we call stage 4 or after they've spread from their local site of growth, we could sometimes extend survival for months, but very rarely cure or have durable cures of these advanced diseases. What immunotherapy has really done is create an extension of survival for some patients and actually a significant increase in the proportion of patients that have long-term, durable, or even curative treatments. But you can see from this curve, which shows it's supposed to represent about a third of patients who are actually deriving this long-term benefit, we still have patients who are receiving immunotherapeutic treatment and either not deriving this really effective deep cures, or if they are having responses, are short-lived. And so the field is still really focused in trying to understand what differentiates these patients who do have long-term responses and durable responses from those that fail to receive benefit. One of our understandings is that while we may have shifted from standard treatment to a short-term survival benefit, we're seeing this fraction of patients with long-term cures, studying really what are the factors, either of those patients' cancers and or their own immune systems or other host factors. We know that many other things influence how the immune system actually responds to cancer treatments, such as, many of you may have heard of the microbiome, which is a very relatively new field in terms of trying to understand how the microbacteria and bacterial, and in some cases even viruses, within our own host influence immunologic response. The microbiome has a significant influence on response to cancer immunotherapy. And we're really just beginning to understand some of the host factors and tumor-derived factors that predict who are these patients who have long-term durable benefit from those that don't. Through trying to then combine approaches, either of immunotherapy approaches or even integration with standard therapy approaches, the hope is that we'll continue to increase the proportion of patients that have these long-term durable benefits and really start to speak about cures in settings where we really previously hadn't discussed the potential for curative treatment. And we're seeing some of this in perhaps some unpredictable or unanticipated ways. So chemotherapy, which has been a long stay in the treatment of cancer, and we know does have negative effects on the immune system. Typically, there's certain cycles of chemotherapy. People's white cell counts oftentimes drop. They can become more susceptible to infections, and then the body does seem to recover between these cycles. We're finding that combining chemotherapy and immunotherapy in integrated fashions can actually have additive or even sometimes synergistic benefits. And so we're trying to understand why we may have predicted that chemotherapy might limit some immunologic responses. In some cases in lung cancer, as an example, the combination, and even in triple-negative breast cancers, of immunotherapy and standard treatments is actually looking quite effective. And so this is an active area of research within our own field, as many others. The other thing that people have had concerns about is, well, what about the side effects of immunotherapy? So if you take the host's immune system, I mentioned to you, many of these regulatory signals are there to prevent us from attacking our own bodies with the autoimmune disease or long-term inflammation. It turns out that some of these treatments, particularly the ones that block those negative regulatory signals, can have side effects that we call immune-associated adverse events. And so there's been a lot of study in looking at what is the side effect profile of standard immunotherapeutic treatments. Most of this, although not all of this data, comes from the use of immune checkpoint inhibitors or a form of T-cell therapy where the T-cells are expanded outside the body and genetically engineered to recognize cancer cells more effectively. They can lead to a spectrum of side effects that are actually shown in this diagram from the National Cancer Institute. The size of the blue dot actually indicates how frequent overall these side effects are actually seen. And then the red dot determines the severity in terms of where do we see more severe symptomology. And so you can see side effects really that impact almost any system within the body, but there are some classic groupings of areas where we see either inflammatory responses or even autoimmune disease that can be induced by the treatment that is being delivered to these patients. Fortunately, in the majority of patients, these are mild to moderate and reversible symptoms. that can be managed with vigilance and also sometimes a break in treatment therapy and also the use of corticosteroids. And oftentimes these patients can actually resume with treatment once those immune adverse events are controlled. And there have been now large-scale studies of comparing, well, how does immunotherapy compare to conventional treatments? And so some meta-analysis that have looked at tens of thousands of patients, particularly from randomized clinical trials where they were receiving either immunotherapy or chemotherapies, have shown that the cumulative side effect profile of patients that have significant side effects that at least require medical management is about 16.5%. That's a broad range. I mean, there's a broad range with those numbers, but that was the average from one of the larger meta-analyses. By comparison, if you look at chemotherapy treatments from those same studies, the incidence of side effects that at least required some medical management was actually 40%. So while these are a new spectrum of side effects that the field has had to learn to recognize and treat, comparatively speaking, immunotherapy has actually been a well-tolerated and effective treatment regimen to integrate into cancer care. So for the last few minutes, I will touch on efforts in brain tumor immunotherapy. So brain cancer, although not the most common of malignancies, are actually one of the most challenging cancers in oncology due to the fact that, of course, all of our organ systems and the essence of our memories, personalities, communication skills, et cetera, are all housed within the central nervous system. In adults, the most common malignant brain tumor is glioblastoma, which is a stage 4 glioma, meaning that unfortunately the majority of patients present with a disease stage that is already advanced and can't be cured with standard therapies. Standard treatment for this disease involves as much of what we call gross total resection to try to remove with surgery as much of the tumor as possible. Even though the tumor has spread beyond that site, we know that removing a lot of the tumor burden can reduce, can extend survival, but also reduce the symptoms that some of the tumor pressures and swelling can inflict on patients, and also improve their quality of life. Then they typically have a radiation and chemotherapy combination that extends for anywhere from 6 months to a year post-surgical resection. And a more recently approved, FDA-approved therapy is also using alternating electric fields that can actually slow tumor progression. Despite this very aggressive treatment and duration, most patients unfortunately spend the rest of their lives going through this treatment with a median survival of only 15 to 18 months. So there's a real significant need to try to improve care options for patients with glioblastoma. Pediatric brain tumors are actually less common than adult tumors, but have actually become now the leading cause of cancer-related deaths in children. And that's really because of the remarkable advances that we've made in treating more common childhood malignancies such as leukemias. Standard treatment also, we do cure more children with malignant brain tumors than we see in adults on average. But because of the developing nervous system and the sensitivity to some of these standard treatments, particularly radiation, but even chemotherapy on the systemic and developing organ systems, the treatment for those children that do survive is very harsh on the developing nervous system as well as other organ systems. And then for those that do suffer relapse, about a third children that have relapse after radiation, these tumors are almost uniformly incurable at that stage. And so our center has focused, and the field is focused, on trying to determine whether immunotherapy can be an effective and safe treatment for both childhood and adult brain tumors. So this is a current snapshot of the University of Florida's Brain Tumor Immunotherapy Program clinical trials. These are actual treatment approaches that have been developed through laboratory investigations by our faculty and have progressed through the stages of preclinical evaluation, what we call FDA, investigational new drug enabling studies, and then have received FDA approval to move into first-in-human clinical trials. So these are discovery to intervention immunotherapeutic approaches. And you can see that our center is really pursuing a wide variety of approaches to treating malignant brain tumors, including cancer vaccine approaches, adoptive T cell therapy approaches, This year we'll open a new form of therapy using actually stem cells from the bone marrow to actually reprogram the immune response against malignant brain tumors, and have several trials that are looking at how to make immune checkpoint inhibitors more effective in the treatment of brain tumors. These actually approaches open the blood-brain barrier to allow these checkpoint inhibitors to actually get into the tumor microenvironment, and also increase the release of tumor antigens, and in some cases have been shown to be effective for these patients. So one of the earliest approaches that we've developed in our center was actually based on this understanding of the role of dendritic cells and their importance for activating the immune system. And our lab has been studying RNA-based vaccines long before COVID, and everyone became very familiar with RNA-based approaches, but using messenger RNAs that encode for tumor antigens, and then either loading them into dendritic cells that have been harvested from a cancer patient, so that the RNA is then converted into the tumor protein. The dendritic cell processes that protein to present it to the T cells of the immune system, and then we return these loaded and activated dendritic cells back to patients that are bearing cancer, with the hope that they will migrate to the lymph nodes and induce that program that I showed you in the cancer immunity cycle. We've also recently and just started a first-in-human clinical trial where the RNA is directly injected into patients, similar to in a formulation to what the COVID mRNA vaccines work, and then these are taken up by dendritic cells in the patient's body and used to stimulate immunologic responses. That trial just opened a few weeks ago and has treated its first patient with glioblastoma. And then the other approach that we have used is using this same paradigm to use these dendritic cells to activate the T cells outside the patient's body, expand those T cells to very large numbers, larger than what could be achieved inside the patient's body, and then return the activated T cells as an adoptive T cell therapy approach. So our group has centered on the use of RNA-loaded dendritic cell vaccines for either vaccine priming or T cell therapies for many years now, for well over a decade and a half. This is actually the results of the first phase 1 study that ran with our colleagues at Duke, where we looked at patients with glioblastoma who received standard of care treatment. So all of them went through surgery, radiation, combined with chemotherapy. But then we integrated a dendritic cell vaccine made from their own, taken from their own white blood cells, loaded with RNA, expressing a tumor-associated protein, and then returned back to those patients during each intervening cycle of chemotherapy. And these patients were actually randomized to receive one of two dendritic cell vaccines. The first was combined with a tetanus booster shot, and the second were actually did not receive the tetanus booster. And what we were studying at the time was whether or not if you induced a strong immune response against something the body had seen already, like tetanus, would that actually make the dendritic cell vaccine have a more effective response because the body would see it in the context of a memory recall response or immunologic response. And so patients were randomized to receive one of two of the dendritic cell formulations. We also studied where those dendritic cells go, but in the interesting part of the study was that those that had received the combination tetanus toxoid booster along with their dendritic cell vaccines actually had more than double the overall survival of the other patient cohort group. And we actually had 50%, it was a small study, but 3 of those patients that were randomized on this arm actually had survivals that are now out past 5 years. And so this was a small but significant response that had a very interesting underlying mechanism of action that we also discovered during this treatment. And this has now moved on to a large randomized phase 2 trial of 150 patients that's recently will complete enrollment at the University of Florida on that study in the coming months. So this year we will hopefully determine whether or not this has been replicated at a much larger scale now with a large number of patients. This is actually an MRI from a patient that's on this phase 2. It's called the ATTACK-2 clinical trial. This was a gentleman who was diagnosed with a midline glioblastoma. The area of white that you see on this scan is actually the normal brain appears gray. This is actually glioblastoma tumor. And because of the location of where this tumor grew, this patient could not have a complete resection. So post-surgery, he still had quite a significant amount of disease. And so what you're looking at are slices in the MRI as the image is moving through, and every area of white enhancement is an area of glioblastoma that was left behind. This is a pretty poor, what we would call poor prognosis when you can't resect the tumor, and also based on its location. This patient did have standard treatment along with these integrated dendritic cell vaccines. And after his 5th vaccine, actually had a complete radiographic response to treatment. And it's been actually now, this July will be 4 years that he's been tumor-free. And so we're excited about the potential for patients, and this is obviously a pretty significant and dramatic response, and we'll find out when the study is completed in a large population cohort whether it has had a meaningful benefit to the patients who have received these vaccines. This is a picture actually of the second patient that was enrolled on the phase 1 study that we started in 2006. Her name is Sandy. She's been featured on television a couple of times. She was 61 years old when she was diagnosed with a glioblastoma and told that she had about 9 months to live. This is a picture, I think, taken in 2015, which was 9 years post-diagnosis. And she is currently now approaching 16 years tumor-free from the phase 1 study. So there certainly are some patients that we believe have a meaningful benefit from cancer immunotherapy treatment, even with advanced brain cancers. However, we don't see complete responses in all patients that are treated, and we'll find out whether on a population level we've had a meaningful benefit to a large cohort, but we are still actively trying to understand, just like others in the field, what differentiates these remarkable responders from patients who don't respond. Another form of therapy that we've worked on is this adoptive cell therapy platform I described to you, where instead of activating the T cells in the patient's body, we actually do this in basically in a pharmaceutical-grade clinical laboratory called a GMP facility, and then return these activated T cells back to patients. This is an example of a child with metastatic medulloblastoma that also had a complete response to adoptive T cell therapy. The two figures here are showing the tumor in the cerebellum as well as in the cortex's brain, where all the areas of white that you see are actually tumor cells that have spread essentially throughout the entire CNS of this patient. He also unfortunately had metastatic disease that had spread outside of the brain and even to this thoracic cavity and bone marrow. He received adoptive cell therapy after salvage chemotherapies where the tumor had progressed during chemotherapy and actually had a complete response to treatment. You can see all these areas of white are completely eradicated. Unlike Sandy Hilburn, who's 15 years out, however, disease-free, this patient did not have a long-term durable response. And so we are studying and trying to understand, again, in a case where an immune system becomes activated but doesn't maintain this disease remission, either what has the cancer done to adapt or what has happened with the immune response that has made it short-lived compared to these patients that have these long and durable complete remissions. So in the last part, I'll talk about our efforts to build better T cells and enhance the function of the immune system so that we can hopefully increase the proportion of patients who can derive benefit from cancer immunotherapy. So one of the things that I mentioned to you is this concept of memory and having long-lived immune responses. And this is something that I think has been really highlighted probably to the public awareness by the COVID vaccines. I mean, how many of y'all thought when we had the first vaccines come out, you were like, oh, thank God, we can finally get this behind us. And then you were told you needed boosters, and you're like, okay, I'll get a booster. Then you were told maybe a third one, and you're like, well, wait a minute, how come I've had measles, mumps, and rubella shots when I was a kid, and never had them after that. And so this concept of how durable, even when you've boosted an immune response, is really an important conceptual framework that obviously virologists and people working in the infectious disease setting have been tackling for many years. But in our understanding of what establishes memory against cancer and how the immune response can be re-engaged if those tumor cells either remain or continue to grow is really at very early stages of understanding. So we have worked on increasing the proportion of these T cells that actually have the phenotype of memory cells and these long-lived immune cells. And that has been shown, certainly in preclinical studies and in some studies of adoptive T cell therapy, the more memory-like immune cells that you can give to a patient, for at least in the case of melanoma, the better responses they can have and the more durable those responses are. So that's an area of ongoing effort. We have a new FDA-approved license to investigate a new product where we've enhanced the memory T cells over what I showed you in the prior MRIs. Another area of major research in our center is understanding what are the unique components of a patient's own tumor that are actually being recognized by their immune system. And so we're using computer-based algorithms, some machine learning techniques, and also functional testing to actually identify the antigens that are expressed in a patient's own cancer, and then stimulate their immune response with a patient-specific cocktail, if you will, of antigens that are specific for their own cancer. It's important, you can activate these immune cells, but if they don't get to the tumor or don't infiltrate the tumor, they obviously can't mediate their killing functions. And so using both nanoparticle-based treatment approaches and genetic engineering approaches, we're looking at how you can both follow where these immune cells go in vivo after you give them, and can you engineer these cells to actually traffic better to the tumor microenvironment. And then finally, in an area where we're collaborating with engineers at our university to really understand what happens to these T cells when they enter this hostile tumor microenvironment, and can we, through studying how T cells interact with the immune cells, improve their function so that they can get there and mediate more effective killing. And for this, we're actually using, we're collaborating with an aerospace engineer named Greg Sawyer, who's pictured here. He actually has worked on and still and for many years space exploration and other aspects, but he also uses soft matter tissue, what we call 3D printing of live tissues or soft tissues, to actually study in three-dimensional spaces how these complex microenvironments can be dissected to its individual components. And so we've been working with Greg's laboratory to 3D print tumor cells, 3D print immune cells, and actually study their interactions under high-resolution microscopy, and then compare if you manipulate either the microenvironment or the immune cells in certain ways, how can you make them more effective in killing cancer cells. So this is an example of a 3D printed glioblastoma from Greg's laboratory. This really highlights the challenge of treating this cancer. It was printed, and you saw it started off as a spherical ball, but this is just over a period of 48 hours. You can see these tumor cells that are migrating away from the cancer mass. And so when someone's diagnosed with this brain tumor, the surgeon can take out this central mass here, but there are these tumor cells that in many cases have migrated all the way to the other side of the brain that we have to figure out how to really get rid of. This is actually 3D printing of T cells that have been engineered to recognize the glioblastoma. So the T cells are in green, the glioblastoma tumor cells are in red, and these are what are called CAR T cells, or chimeric antigen receptor T cells. So they have been modified with a receptor that has very high affinity and recognition of a protein that's expressed in these tumors. And you can see once they start to recognize the tumor, they just sort of swarm it, and you can even see they start to dissolve. You can look at the center of this tumor mass as being completely obliterated as the tumor is being chewed up by the T cells. What we're able to do in Greg's laboratory is actually you can go in and sample. Some of these T cells you can see don't seem to really engage. And so what's the difference in these T cells that are sitting out here versus the ones that really are trafficking? And begin to understand what are the components of the biology that really generates an effective T cell, and can we either change the way we generate these cells or modify them in a certain way that they are much more effective at killing tumor cells. So what does the future really look like for this field? One, I think we're encouraged by the advances in the field of cancer immunotherapy in general, that there are approaches that can be effective even for challenging tumors like brain cancer. We are using a lot of integration of large data sets that are coming from other fields to understand some of the factors that predict responsiveness or resistance, and then using that to actually guide our immuno-oncology approaches. The precision immunotherapy platform is really what I mentioned, which is understanding that each patient's tumor is still unique, even patients with the same diagnosis, and how can we use the underlying genetics and biology of their tumor to really design personalized immunotherapeutic treatments, either the right combination of treatment drugs and or the right engineered cell therapies that are recognizing that patient's specific cancer. And then really combining these approaches. Most of these therapies right now are being tested for their safety and their effectiveness in isolation, or maybe integrated with standard treatments. But we haven't, we're approaching what we are really doing are now rationally developed combination approaches based on some of the underlying biology, where we can combine these treatments for hopefully more effective approaches. So in summary, I think cancer immunotherapy is certainly a paradigm-shifting approach to treating advanced malignancies. It's been shown to be a feasible, well-tolerated, and certainly shows promise in some patients with malignant brain tumors. Our future efforts really in overcoming resistance and evaluating combination treatments are warranted and are underway at our center and many others. And I'd like to thank the members of the Brain Tumor Immunotherapy program. This is a big team effort, and many of them aren't pictured here, but this is a pre-COVID snapshot, obviously, of the team. We're working on getting a post-COVID get-together picture pretty shortly. This is our Clinical and Translational Science Institute, where many of our patients actually receive their vaccines in our Clinical Research Center. This is another site on our Orlando campus. Obviously, our funding agencies that have supported this work, and most importantly, the patients and families who've actually participated in these clinical studies and really entrust us with their care, and oftentimes know that they're participating in studies that may not benefit them, but will hopefully benefit patients in the future. So thank you for your attention. And hopefully we have a question in the back there.
Dr. Mitchell, welcome to Pensacola.
Thank you.
In 1938, Dr. Otto Warburg said that one of the cancer's main functions was to protect the pancreas. Do your glioblastoma patients have an elevated A1C as a rule?
That's a really good question in terms of, as you know, the metabolism and understanding really cancer cells' dependence really on glycolysis and whether we can exploit that to inhibit tumor growth has been an active area of investigation. A1C is oftentimes not elevated in patients with glioblastoma as a characteristic of their profiles, but both due to standard treatments, corticosteroid treatments, we do see significant metabolic alterations in patients with glioblastoma and other brain tumors. And actually, glucose restriction and exploiting metabolic vulnerabilities is an area of active research. I didn't touch on that in my talk here, but is an area that actually is under rigorous exploration at our center. And as you can imagine, the interface between metabolism of how immune cells and what nutrients they're dependent on, and the competition for those nutrients in the tumor microenvironment plays a big role in how effective and long-lived immune cells can actually be once they enter the hypoxic and metabolically restricted environment of tumor cells. So this is something that's a real active area of research, but we don't necessarily see that the GBMs are altering the systemic metabolic profile in patients. Sure. We have, I think, back here and then up front. Yep. I think she had her hand raised.
Yes, with regard to brain cancer diagnosis, is there any noted direct correlation between the diagnosis and a compromised blood-brain barrier? And are there presently any reliable tests and or symptoms that physicians or patients can figure this out at an earlier stage?
Yeah, it's a really great question. We had a good discussion about this earlier. So let me touch on the second part first, which is, early detection and possibilities for early stage diagnosis. So there's a lot of active research being done, particularly to see if there's some evidence that there are peripheral biomarkers that may be detectable with proteomic approach, basically looking for peptides or protein signatures in the blood, excuse me, that are indicative of a disruptive blood-brain barrier and or disease pathology within the central nervous system, and quite a bit of research to determine whether or not these are picked up much earlier than we typically diagnose patients with glioblastoma based on symptoms. So some studies that have shown there are altered proteins that can be detected in these patients in the periphery, and a few that actually had retrospective samples that have shown that some of these altered signatures were detectable in patients with glioma months and even years before diagnosis. Whether this is a broad biomarker that can be used for early detection is still being investigated, but it's an area of great interest. When these tumors grow, the chances are that the breakdown of the blood-brain barrier itself is probably at a point at which the tumor has either invaded and reached a certain size, and or often what has happened is the tumor has induced angiogenesis and neovasculature, new blood supply into a growing tumor mass. And that irregular vasculature is actually the leakiest part of the tumor. So what we end up seeing on the MRI in that central mass is often vasculature that has really been developed to feed the tumor itself, and it is more leaky than the classical physiologic blood-brain barrier. So it's not clear how early at the stage of development we may be able to pick up BBB permeability, whether that would be an early stage or a late presentation in some of these tumors. But there's great interest in seeing if either brain-specific proteins or even, and perhaps more likely, alterations of other peripheral biomarkers that are being affected by the alterations that are occurring in the brain may be detectable in the periphery. So it's
We have time for one more question.
We had one other.
That was going to be my question, so since you already answered it, I'll ask you this. Are there any symptoms of early brain tumor?
Yeah, so brain tumors do present with a spectrum of symptoms that unfortunately are not always very specific for brain cancer. In fact, as you can imagine, depending on the part of the brain in which these tumors arise, and they essentially can arise in any area of the brain, headaches can be a common presentation, which is most often not a brain tumor, but is a common spectrum of symptomology associated with brain tumors. Seizures can also, so focal signs of either cognitive, motor deficits or seizures that can lead to imaging much more rapidly and an earlier diagnosis. In children, because of increased intracranial pressure, oftentimes nausea and vomiting, particularly projectile vomiting, can be, again, a particular symptomology. Unfortunately, because we don't have direct pain receptors in the central nervous system, the majority of these tumors, when they lead to symptoms that actually result in imaging and the diagnosis of a brain tumor, they've either arisen in a very critical area, so they're detected still early, or they've gotten to a size where they're a little bit late stage in development. So we don't have great early diagnosis or early symptom presentations for brain tumors at this point.
On that note, let's thank our speakers.